In this video, I'll give you some tips for the post-lab assignment for Lab 6, the milk experiment. Most of the questions on this post-lab are pretty similar in terms of what you have to do for your calculations. In question number 1, this is going to contain data from part 1. You'll be reporting the mass of your test tube. This is the mass of the test tube when it is empty. And then you will be reporting the mass of the test tube with the milk in it. And you'll be taking the difference of these two to get the amount of milk that you used in the experiment. So this is going to be your test tube. I'm just going to abbreviate it to test tube. That's the number that you're reporting right here. And the amount of milk, which is what you're trying to figure out right here, is going to be equal to the mass of the test tube with the milk. And we've done calculations like this before. The test tube and the milk, which is right here. And you'll do the same thing down here for an empty watch glass, a watch glass with casein and butterfat in it, and then give you the total mass. Question number 2, you're going to do pretty much the exact same thing for this part. So there's a lot of this where you are measuring the mass of an empty piece of glassware, and then you're measuring the piece of glassware with something added to it, like in this case it's whey. And then you'll use that to help you calculate the mass of whatever that substance might be. Now in this one, you also have this other question. You're going to report the color of the solution after doing the burette test. And then that's going to make it possible for you to answer this question. Is whey a protein? That's going to be a yes or no. And this is based on the color that you get when you do the burette test. Blue means that no, it is not a protein. Purple means that yes, it is a protein, and hopefully you don't get a bluish purple color. Do your best to figure out what it is. Then you'll do the same kind of thing for these other substances as well. You'll just be doing a lot of weighing an empty dish, weighing it with something in it, and then using that to calculate the mass of the substance, and then 2 more burette tests as well. Now on question number 5, this one is a little bit different. So here you are going to be recording or reporting the total mass of milk that you used in the experiment, and this you calculated back in question number 1. So let's go back and look at that. That's going to be this number right here. You're just going to copy it into this spot. And then here, the mass of all of the isolated proteins. So to get this number, you're going to go back and you're going to look at all of these questions about the burette test. Is it a protein? Yes or no? And you've got one more right there. Is it a protein? So if you answered yes, for example, if you said yes, whey is a protein, then you would take the mass of whey and you would include it in this right here. And you might have more than one substance that is a protein. Like maybe you said that yes, butterfat is a protein, and you would want to take that mass and include that in this as well. So this is going to be the masses, or just one, it might be one, it might be more than one, of all of the proteins that you isolated. And you're deciding whether or not they're proteins based on the results of the burette test. Experimental percent by mass of protein, that is going to be your mass of your isolated, I'm just going to abbreviate it, iso proteins that you calculated in the problem right up above, and then you'll divide that by the total mass of milk, multiply it by 100 to make sure that it's a percent, that's a 1, and you'll also report on here the percent by mass of protein reported on the nutritional label, and these numbers should be relatively similar. They're probably not going to be exactly the same, but they should be relatively the same. You'll do the same type of thing down here for the last 3 questions. So this one is asking you for all of the isolated fats. These are going to be things that are not proteins. So we're isolating in this experiment all the proteins and all the fats. And if you got a negative burette test result, if you decided that no, this is not a protein, then that means that that substance is a fat. So you'll copy the masses of all of the fats into this spot. You'll do calculate your percent by mass of fat. That's going to be pretty much the exact same equation as what you used here. So I'm just going to copy it down into this spot. I'm going to try to copy it down in that spot. And instead of the masses of the proteins, you'll be using the masses of the isolated fats. And then again, you'll be comparing this number to the number that is on the nutritional label for the milk that we're using. And these should be pretty similar, probably not identical, but relatively similar.